Welcome to the FRAXA Research Foundation webinar series. Today we have a much anticipated webinar on the use of EEG as a biomarker for diagnosis and outcomes in neurodevelopmental disorders. By way of introduction, let me just say that FRAXA has started this webinar series, and partly at the suggestion of Dr. Patricia Cogram, so thanks for help in organizing this, Patricia. This webinar series is designed to be a kind of replacement for all the meetings that we've lost. We feel that there's a great need in the Fragile X field for some way of conveying all the fabulous discoveries that have been made in the last couple of years, and we were all very much upset that the Gordon Research Conference in June of this year was canceled for obvious reasons. And we're not sure when we'll be doing big meetings again. But in the meantime, this is the way to get the word out. I'm your host. I'm Dr. Mike Tranfaglia, and I'm the medical director of FRAXA Research Foundation and chief scientific officer as well. So I'd like to say a little bit about the format for today's presentations. We'll be having two speakers, and we'll be taking questions at the end of the webinar, at the end of both presentations. And you can submit your questions via the chat function on Zoom. So please don't use the chat function for just chatting, but you can feel free to get your questions in the queue, and we'll try to take them in the order that they go into the chat. This session will be recorded, so you can watch it on the FRAXA website afterwards, and we're also live streaming on YouTube now. So this is going to be seen by lots and lots of folks. So as I said, today we're going to be talking about EEG as a biomarker in neurodevelopmental disorders. And our first speaker is Dr. Chuck Nelson, who's a professor at Harvard Medical School. He's someone who needs no introduction because he is a leader in this field and is internationally renowned for his work in EEG and human electrophysiology and the study of neurodevelopmental disorders. And so without further ado, I'll turn things over to Chuck Nelson.
Thank you very much. Let me just share my screen. So to set the stage for what you're going to hear today, I'm going to provide an overview to what we mean by biomarkers, how biomarkers can be used in neurodevelopmental disorders, and then talk a little bit about trajectories to autism. And then Dr. Wilkinson's going to come in and talk about some of the exciting work she's doing more recently on EEG and Fragile X specifically. So let me, okay. There we go. So let me provide some context for how people have used this term biomarkers and the kinds of studies they've used biomarkers in. So biomarkers can be used as an endophenotype or an intermediate phenotype, that is to link genes with behavior and to sort of dig beneath the surface out of range of behavioral assays. So something that, in a sense, sits beneath the way we can observe behavior. For some reason, there we go. It can also be used to shed light on mechanisms. So work that Mark Bear and I are doing, for example, is going from mouse to human using the same assays, in our case, EEG and the visual evoked potential. They can be used as a predictor or as a characterizer, a word I just made up. So we can use it to distinguish individuals who are at high risk or at low risk for a disorder, to predict who might develop a disorder, which is what I'll turn to in about 5 minutes, and maybe to even develop a way to predict the severity of a disorder. They can be used as a moderator or a mediator in clinical trials. So you can use biomarkers to stratify a sample, rather than say stratify based on disorder or not a disorder or level of intellectual functioning. You can use the biomarker to stratify a sample, to examine a response to treatment, and to shed light on neurobiological mechanisms that underlie treatment efficacy. So in work we did some years ago with Rett Syndrome, we were using EEG not only as a readout for the efficacy of, in this case, IGF-1, but as well to see if we could maybe shed light on how IGF-1 was impacting synaptogenesis. So what are some of the considerations in biomarkers? Well, Can they get as close to the mechanisms that underlie the disorder, that mediate response to treatment? Can they be tolerated by, in my case, infants and young children? I'm less concerned with adults. Expenses another consideration. If we're going to roll these out in large-scale clinical trials in multiple locations, it can't be something that's very expensive. Can the effects be replicable across labs? Can they be replicable across individuals? And when they're used longitudinally, can they distinguish changes in the disorder from developmental change? And this is a non-trivial issue, because if you follow the same individual or a group of individuals over time, how do you know that the changes you're seeing are due to the treatment rather than just changes in development? So the work I'm going to talk about today is now grounded in the study of autism, with the premise that early detection of autism can lead to earlier treatment, and earlier treatment can in turn lead to improved outcomes. So we're going to, we're more broadly drawing on the field of cognitive neuroscience. And currently, autism is a behavioral diagnosis, but we need to, if we're going to detect very early in life, we either need to wait for behaviors to become more sophisticated. In other words, for the behavioral repertoire of the child to become sufficiently advanced that you'd have a reliable predictor of later autism, or you focus on behaviors that do appear very early. Eye movements would be an example, turning to name, eye contact, things like that. But the other option, of course, is to focus on the brain and bypass behavior entirely. So we assume that changes in the brain precede changes in behavior. Can we assay the brain directly, bypassing behavior, or in the case of, that I'll talk about today, study changes so early in development, there really are no behavioral readouts possible in development yet. So there are, in my case, my interest is in predicting who will develop autism. And to do that, we usually draw from an enriched sample. These are children at elevated risk for developing autism. And it turns out that there are a large number of types of disorders that confer increased risk for autism. So there are those with genetic syndromes like tuberous sclerosis and Fragile X, Rett syndrome, Angelman syndrome, Bailey-McDermott syndrome, Down syndrome. There are copy number variants like 16p11.2, Duchenne, even Duchenne muscular dystrophy. There are, of course, also those who have an elevated risk for autism based on familial risk. So low birth weight or prematurity, ventricular megaly. Turns out infants raised in institutions, infants with an older sibling with autism, which will be the focus on my remarks starting in a minute or two. And there are two things we don't know about all these populations. So the first is that, given the many pathways that lead to autism, you could ask, is this the same disorder or variants of the same disorder, or are they possibly different disorders? So when you have a fever, you don't have a fever disorder or a fever syndrome, but fever represents different underlying conditions. So the question is, we're making a diagnosis of autism based on behavior, but if we more finely splice this, as it turns out that are there different variants of autism, or maybe they're completely different disorders. And then the other question is, what leads some children to develop autism and others not, given comparable risk? So if we go back to the previous slide here, in some cases, I assign the risk that a child with a given disorder will develop autism. So by some estimates, 60% of children with tuberous sclerosis develop autism, but that means 40% don't. So the other way to think of this is not just who's at risk, but who's at risk but doesn't develop the disorder. So we're going to talk today about one specific population of children at risk, and these are babies with an older sibling with autism. And this work is not necessarily, this problem rather, is not a new problem. The BabySib Research Consortium was funded 15 or 20 years ago. It represents more than 18 groups around the world, and the focus is on the outcome of infants who have an older sibling with autism. And we know from sort of the epidemiology here, that about 20% of babies who have an older sibling with autism will eventually go on to develop autism. To date, there are no behavioral measures in infants under a year of age that reliably predict autism outcomes. There is a little bit of work from Ami Klin's lab from eye tracking in infants less than 6 months, but that's sort of the exception to the rule. So we have to assume that there are no behaviors in the behavioral repertoire below a year that reliably predict autism. So here's a way to characterize this. So of 5 children, 1 will develop autism, 1 might develop a speech or language delay, 1 might have a problem with anxiety or ADHD, and then 1 or 2 will be typically developing. But in that context, we should add that we actually don't know the long-term outcomes. I'm sorry, my bandwidth is such that it's taking me a while to advance the slides. We don't have any long-term studies. So if you look at the molecular biology of autism, the disorder that most closely aligns with the genes that have been identified as playing a role in autism are those involved in schizophrenia. But these are very, very different disorders, but we have no studies that go out 30 years that have tracked infant SIBs. So one can only ask, will a baby who is typically developing at 2 or 3 still be considered to be neurotypical 15 or 20 years later? So the project I'm going to talk about today has been done in collaboration with Helen Tager-Flussberg, a colleague at Boston University. And we focused on 2 groups, high risk for autism by virtue of, excuse me, having an older sibling with autism, and then low risk children. These are children with no family history of the disorder. And we expect about 1% or 2% of these children to develop autism. The way the study works is that we begin to collect data at 3 months of age, and then we follow the children again at 6, 9, 12, 18, 24, and 36 months of age when we can make a definitive diagnosis of autism or not. This is a big commitment on the part of families because it means they're coming into the lab many, many times for us to follow their development. And we already have to acknowledge that the parents who already have a child with autism are just wondering what the chances of their infant developing autism. So we have the 3 groups then eventually. We have those who have a high risk for autism who eventually develop autism, high risk for autism, but who wind up to turn out to be typically developing, and then the low risk typically developing children. So by way of background, in this one study, we've actually now have a new cohort, but I'm going to focus on our original cohort. We enrolled 220 participants. We eventually had got ADOSs on 174 of them. 72 were low risk and 102 were high risk. Excuse me, being on Zoom all day, I'm already losing my voice. And of the high risk, 30 of them developed autism. So in our cohort, roughly 30% of our high risk infants eventually met criteria for autism, which is markedly higher than the BSRC figures of roughly 20%. In addition, 55 were males and 47 were females. So in this case, males were 2 times as likely as females to develop autism, which of course is also different than the standard figure of a 4-to-1 ratio, which you see in idiopathic autism. So it's possible, and we may come back to this later, that infants at risk for developing autism who then develop autism develop a different version of the disorder than those who don't have a sibling with the disorder. So here's what we're going to focus on today, which is the EEG. When the lab is open, Dr. Wilkinson will attest to this, I spend all day blowing bubbles trying to keep babies busy. And here we have a team of folks wearing our EEG nets, and here's a budding neurologist doing a read of an EEG. And what we do is we record the EEG for several minutes during these baseline conditions. The advantage is that EEG is non-invasive, it's relatively inexpensive. The same tool can be used across the lifespan. So what we do with a young baby, we can also do with an adult. It's very well tolerated by nearly all children. It places minimal demands on language and attention, and it has excellent temporal resolution. Disadvantages, well, it doesn't have very good spatial resolution. The scientific promise is that we use it neurobiologically to draw inferences about the activity of large populations of neurons that are acting in concert with one another. And we may be able to provide a link between what we see at the level of a synapse and what we see at the level of behavior. So here again is our way of keeping the baby occupied. So, here's the power spectrum of the EEG. We have a multiplex signal, and we can decompose that signal into different frequencies and measure its power spectrum, which you see here. And here's a way to look at that decomposition, where we can break the frequencies into very, very slow activity all the way up to very, very high activity. And higher power suggests multiple neurons are oscillating synchronously at a given frequency, and these field potentials are enough to be detected at the scalp surface. So let me go through some findings. And the first part of the data I'll talk about today were overseen by April Levin, a child neurologist at Children's, and Candace Farson, a former postdoc who's now back in Australia. So in this longitudinal study, we began by doing something very simple, which is just looking at different frequency bands and power. And the first pass was to look at whether we could distinguish the high-risk children from the low-risk children at 3 months of age. And what you can see in the lower left is for high alpha activity and for beta activity, the children at risk for developing autism have lower power than the children who are low risk for developing autism. But remember, we haven't broken the sample down yet into those who do and don't develop autism, which is what you'll see in the next slide. So now, if you look here, in beta and the gamma band, the high risk for autism who develop autism have even less power than the high risk who don't develop autism, who in turn have less power than low risk. So what that's telling us is that at 3 months of age, we can distinguish the infants who later develop autism from the infants who don't later go on to develop autism. So the next thing we tried to do was to make better use of our longitudinal data. And so this work was overseen in part by Laura Gabbard-Durham, a postdoc in the lab, and Carol, and several others. So what you're looking at here is the the EEG collected from 3 months all the way to 36 months. And we're going to have a group of 31 children who developed autism by the time they were 36 months. Another group of children who did not develop autism at 36 months, but had high risk for developing autism, and then typically developing children who were at low risk and did not develop autism. And so we're going to look at the change in the first year of life versus the change in the second year of life. So the way we did this is we looked at the sum power in each frequency band, we computed growth trajectories, and then we looked at the slope and the intercept over time. And this is the beauty of doing longitudinal studies, is that we can look at change from 3 all the way to 36 months. So what we're going to ask is, do we see a difference between those at high risk who develop autism versus those at high risk but don't develop autism, or those at high risk who develop autism versus those who are typically developing and at low risk? And here's what we see. In the first year of life, which is the darker shade of blue, we have an accuracy that approaches 90% for distinguishing those at high risk and develop autism from those at high risk who don't develop autism. And similarly, a little bit more than 90% for those at risk for developing autism and who develop autism from those who are at low risk and are typically developing. You'll notice that the accuracy is much higher in the first year than it is in the second or the third year. And I'll come back to this in just a minute. The next part was to look at the positive predictive value. So how many of the participants labeled with autism actually had autism? And you can see again, if we do the comparison of those at high risk who develop autism versus those at high risk who don't develop autism, our positive predictive value is above 70% in the first year, but then drops in the second and third year. And if you look at those with autism from those who are typically developing and low risk, we still have a positive predictive value that's approaching 70% in the first year. So the first year was the most accurate in predicting autism. The spatial specificity, which I didn't talk much about, was primarily over the frontal lobe. And so the differences across age, first year, second year, so on and so forth, were greatest for electrodes distributed across the frontal cortex. And these longitudinal changes in EEG power are very informative. So slope measures, that is change over development, were important in all the models. And this, of course, tells us something about where we should be looking. And so what this is telling us is that if we have an infant with an older sibling with autism, examining the development of EEG across the first year of life may be highly predictive of whether that child will eventually develop autism. So now in the last part of my talk, in the last 5 or 10 minutes, I want to talk about some work that really began more than a decade ago by Bill Boswell. Bill is a computer scientist, was a K Award winner, awardee rather, in the lab, and got us interested in the brain as an evolving, adaptive, complex, dynamical system. So let's start with, what do we mean by complexity? One way to think of this is meaningful structural richness. Complexity is not the same as complicated. So complex would be hierarchy of scales, patterns on patterns on patterns, richness, layers, variability, surprising but orderly, like Beethoven's Fifth Symphony. On the other hand, complicated would be our tax return. And there's a difference between complicated and complex. So healthy physiological signals are complex. Here again, we have the continuous EEG, which looks very, very noisy, but we can take this multi-scale, information-rich, complex signal and look at it in a number of different ways. In the first part of my talk, we talked about spectral power, but the reality is that spectral power does not capture the full dynamics of the EEG signal. So we think of the brain structure as complex. These neural networks exhibit a particular kind of structure, which you can see in these two pictures here. The electrical signals produced by the brain are complex, so EEG time series are chaotic, and these things are related. So complex hierarchical networks produce nonlinear chaotic time series that contain information about neural structure. So let me just pause for a second and decompose that a little bit. Earlier in my talk, I mentioned how we're using EEG to look at the neurobiology of brain development. And I think what we're saying is that we're trying to extract features from the EEG, both in time series and in spatial distribution, that we think will allow us to draw inferences about how neurons are communicating with one another and how networks are forming. The principles here are mostly mathematical principles, not so much biological principles, and I'll come back to that when I talk about a few cautions at the end of my talk. So there are a number of ways to look at features of the EEG. By now we have a list of more than 20 of them. There's a few that I'm listing here. The question is, which of these are the most important in detecting the neural correlates of autism? And are some of these redundant or irrelevant? And that's important clinically because it tells us what we really should focus on if we're starting to roll something like this out in a clinic. So we're going to start with this one study that I think is worth paying close attention to. So we begin with these internal dynamic trajectories of different neural states. This would be conveyed pictorially here, but what we want to do is say, okay, so we can take these different neural states, we can capture them in the richness of the EEG, we can compute all the signal features possible, and then take these features, extend it to machine learning, and then from machine learning, draw inferences about cognitive function. So here is using a sport vector machine, one way to do this. So we use this leave one out training set, and then we use machine learning to sort of separate the planes of the different features that we're extracting. And what we see is in blue, these blue squares, these are the separated plane of the low-risk children who don't have autism. The green are the high-risk children who don't have autism, and the red are the high-risk children who went on to develop autism. And the separation you see in this SVM model indicates that we can cleanly distinguish those three groups from one another. The other thing is that, so here's another way to characterize this. The 3-month-old EEG predicts autism to 95% accuracy at 3 years of age. In addition, we can, oops, sorry, let me go back. In addition, we had some success in predicting severity of the disorder. So here we have, the green would be the high risk, no autism, the blue are the low risk, no autism, and up here in this reddish color are the high risk with autism. And so what we find is that we have some great success actually in predicting autism and some success in predicting severity of autism. Now I need to qualify that by saying also that the variant of autism that children who have an older sibling with autism develop tends to be milder. So the range of severity is much more truncated in our cohort. So for example, of our infants who developed autism out of the high-risk population, all have language. We don't see any minimally verbal children, for example. So to summarize, there are now sophisticated ways to capture the nonlinear dynamics of the EEG. Bill and with us have shown that it's possible to predict autism from 3 months of age and possibly also the severity of autism. And combined with our earlier work using more conventional approaches, spectral analysis and the like, to EEG, this opens up the door to development of predictive biomarkers. What are some of the questions that we should be asking? So first, with regard to the EEG as a predictor, we could ask, what is it we're predicting? So we're focused on autism and we find great success in predicting autism, but might this really just be an error in brain development? And if we had measured other things besides autism, we might also see differences in the EEG at 3 months of age. And so this raises the issue of specificity. We have high sensitivity, but if we're only looking at autism as the outcome, we should be cautious and draw conclusions that what we're picking up are signs of autism per se, rather than another disorder. Another is our work was designed to allow us to identify who's at the greatest risk for developing autism and predicting who will develop autism. But we could also use these tools as in intervention studies. But then the question is, do we treat the biomarker or do we treat the disorder? So epilepsy is an example. Do we treat someone who has epilepsy, or do we begin by treating abnormalities or atypicalities in their EEG, even if they don't have a seizure? And so if at 3 months of age, long before there are signs of autism, we see something different in the EEG, do we treat the EEG to normalize it, or do we wait till there's a disorder? The last thing I think is, given how early we observe differences at 3 months, how early does the disorder really start? In my mind, if we're seeing differences at 3 months, this may well have a prenatal origin. And when would we start to intervene to make a difference? So again, right now, usually children who get early intervention services are those who are showing clear symptoms of a disorder or have autism. But if we're seeing differences as early as 3 months in the EEG, what would an intervention look like if it targeted 3-month-olds? On that note, I'm going to stop. I promised Carol I would take only 30 minutes, and I'm exactly at 12:30. And so my memory is that we're going to go right into the second talk and then field questions. Is that right?
Yeah, so thank you very much, Dr. Nelson. That was fascinating, really powerful technology, really exciting possibilities. And yeah, if you have questions, feel free to get them in the queue through the chat function. And then we'll do Q&A at the end of our second presentation. And our second presenter is Dr. Carol Wilkinson, who's a FRAXA postdoctoral fellow and an instructor at Boston Children's Hospital. Carol is a developmental pediatrician, a clinical researcher, and also a basic scientist, classic triple threat. So Carol, over to you.
Well, thank you so much for inviting me to give this talk. I'm very, very excited to share data from our Fragile X study in young boys that was in part funded through FRAXA. And so we really have a lot of thanks to the foundation for that support. First off, I don't have any disclosures. As Mike said, I'm a developmental behavioral pediatrician, as well as a cognitive neuroscience. And so I know most people on the call are very familiar with Fragile X and the different challenges that children and adults have with Fragile X. But I put this here really to emphasize a few things. One is that the challenges that are often faced by children and adults with Fragile X include intellectual disability, language delay, a host of behavioral challenges. And I put these lines connecting them together because I think that we can't think about them as individual things. I think we really have to think about the interconnectedness of those different challenges and how behaviors can affect language, how language can affect behaviors, and how cognitive challenges also can affect those things. The other aspect is that for me, there's a huge amount of overlap in Fragile X with many other disorders that I take care of. So autism, obviously, 30 to 50% of kids with Fragile X will also meet criteria for autism, but a huge number have great difficulties with attention and hyperactivity, as well as anxiety. And so understanding the underpinnings of those challenges for Fragile X, I'm hopeful that that will also shed light on kids that do not have Fragile X, but share some of those challenges. And the last part I want to highlight are these pictures. So these are pictures of same two individuals as young children and then as adults. And you can see that their faces, one of the features of Fragile X is sort of facial elongation, right? And the reason why I've put this up here is to really emphasize the developmental aspect of the disorder. So when we think about, or at least when I think about changes that occur in the brain and how those relate to these different cognitive, language, and behavioral challenges, I think about it from the developmental perspective, because many of these challenges change over time and are the result of a developing brain. And I think that this picture always reminds me of that. And so before going into the study, I thought we could also just briefly go over sort of the history of all of the knowledge we have gained about Fragile X, the molecular mechanisms behind Fragile X. In part, I'm doing this because I think it highlights both the complexity of the disorder and the challenges that still remain. And so almost 30 years ago now, the gene that's responsible for Fragile X was identified, FMR1, and that's a expansion repeat. It's very complicated, it turns out, because it's not just whether you have a mutation or not that leads to lack of production of the protein, but actually also methylation. And so there's complications from the get-go, even from the genetic standpoint. 2 years later, the protein FMRP was described, and it's an RNA binding protein and regulates protein translation of many synaptic proteins. So a broad impact across synaptic plasticity. And then very soon after, the knockout mouse, a knockout mouse was developed. Nicely, the knockout mouse has many overlapping features with humans with Fragile X, including things like hyperactivity, impaired social interactions, some cognitive deficits, repetitive behaviors. They also have increased seizure activity, which is maybe not quite an overlap with humans. Individuals with Fragile X are increased risk for seizures, but they don't have these audiogenic seizures, which are sort of one of the very robust findings in the mouse model, where the mice will have seizures with large sounds. In addition, the knockout mouse has demonstrated many different issues with synaptic plasticity and overall synaptic dysfunction. And you can imagine that would be true because the protein that's involved is not specific to one regulating one protein's translation. It's a host of proteins. And so many receptors have been identified and in a bidirectional fashion affecting synaptic plasticity. And then in turn, we've also seen that there's overall network dysfunction, balance of excitation and inhibition is dysregulated in these knockout models. And so then in the early 2000s, work predominantly by Mark Baer's lab and then followed by others found that mGluR inhibitors, mGluR LTD was found to be one of the pathways disrupted in Fragile X, and mGluR inhibitors were found to improve the Fragile X phenotypes in animal models. And since then, a host of other possible therapeutic drugs have been identified in mice. I think it might be Mark Baer who says that you're doing really well right now if you are a Fragile X mouse, and that we're able to really improve a lot of the phenotype in the animal model. And that gave us great hope. And you can see with this list from Liz Berry-Kravitz's article in Pediatric Neurology, all the different potential candidates that are either in preclinical all the way to phase 2 or 3 clinical trials. And those clinical trials started in 2009. And the frustrating part about it is really that we haven't seen great success. There have been several large phase 3 trials that did not meet their outcome goals. And so we still do not have drugs that are FDA approved. Although when we look at the different trials have looked at populations, there has been hope. But this raises some challenges in why we haven't been able to translate what we see in the mouse over to humans. And so what are those challenges, or what do I see those challenges as? So one is timing, right? A lot of the clinical trials have been done in adults or adolescents. And this is, as I talked about in the beginning, a neurodevelopmental disorder. It's a disorder that likely starts prior to birth with changes in the brain that affect, lead to different challenges, cognition, language, behavior, all of those things. And those things may be harder to change in an adult compared to a child. We also know less about the brain differences that are present in children compared to adults right now, because much of the research has been done in adults. And so one goal that we need to address is starting to do these trials in younger children, or children period, and understanding the biology there. Now, the other aspect is heterogeneity. I think many people think of studying Fragile X as a way of getting away from heterogeneity, that, oh, I'm going to study a genetic disorder, so there's, everyone has some degree of intellectual disability, or there's just less heterogeneity. But that doesn't mean that heterogeneity does not exist in the disorder. And in fact, there's quite a bit of range of symptoms within the Fragile X individuals. And that can be on many, many levels. So if we have 5, 6 individuals, they may all have varying degrees of IQ, some in the mild range, some in the severe range. And those same individuals may have different profiles of language. And maybe also their sensory sensitivity may be different, and they don't necessarily correlate. Someone with a low IQ doesn't necessarily have extreme difficulties with sensory sensitivity. They're sort of very heterogeneous in that way, and that happens for anxiety. And then also for the therapeutic response, and that's what I think we've seen in some of the clinical trials, is that not everyone responds to these drugs equally. And identifying who is going to respond to a drug ahead of time may improve our outcomes in clinical trials. And then the last challenge that I want to mention is this sort of lack of outcome measures. I think that several other webinars from FRAXA have discussed this. That right now we really are using parent surveys and different psychological tests as outcome measures, and those have challenges. Parent surveys are difficult because parents are asked to sort of give a value on a child's behavior at a given time, but if you talk to a parent, they will tell you it sometimes fluctuates from day to day, and trying to figure out what the average is over a month accurately can be very, very hard. There's also that placebo effect that can occur. Psychological tests, we've struggled with standardized testing, has not been developed for individuals with intellectual disabilities. So there's often a floor effect. There are new toolboxes through the NIH that I think will be valuable. But the other thing is, do we expect change over short periods of time, right? Do we expect someone's IQ to significantly change over a 3-month clinical trial period? And is there another measure we could use? And that's really where the biomarkers come in. And EEG as well as eye tracking, I think, are 2 biomarkers, along with others that are in the pipeline. And EEG is the one that I'm going to be talking more about today. And so the need from my standpoint, or for what this small study is addressing, is that we really need biological markers that are associated with functionally significant outcome measures like language, cognition, behavioral challenges. We need those both to be used in trials to monitor improvement, but also to improve our understanding of the pathophysiology, right? So that we can also develop additional therapeutics, new therapeutics. And so EEG is one of those potential biomarkers. And as Chuck described, it can be easily placed on infants' scalp, as well as individuals with Fragile X. And I'll show pictures for any parents that are out there and wondering if their child who may have extreme sensitivity to being anything being put on their head, how we approach that. And then we can take those signals and we can do many, many types of analyses. One of those is looking at the different frequency bands and the overall power of the EEG to better understand how that relates to different outcome measures. And one of the reasons why EEG in particular for Fragile X has been exciting is because of the work by the mouse work and human work, both by Khalil Razik's lab. He gave a webinar, I think earlier this summer, if you want to check that out, as well as Lauren Etheridge's lab, who's been working in adults and adolescents with Fragile X, and the similarities of their findings. So both in mice and in humans, we've seen that there's increased power in these higher frequency bands. So this here is what we call the power spectrum. It starts with low frequency like delta and theta and goes all the way out to higher frequencies. Gamma is in the sort of 30 and above range. And the Fragile X mice are this blue line, and you can see that they have much elevated power once you get to those beta and gamma ranges. And that's the same thing that's seen in humans as well. So increased beta power in humans. In addition, both of these groups have also seen similarities in the EEG response to auditory stimuli. This is this chirp paradigm where you listen to sort of a ever increasing chirp that the sound goes from a low frequency to a high frequency sound. And our brains, when we hear this sound, we entrain to the frequency that's heard. And so you see increased power, which is like in red or green, as the sound is increasing in frequency. And what we see, we see in the mice is that there is a decrease in that sort of entrainment in the gamma range. And Lauren Etheridge's group has seen very similar findings in adults. So really promising, I would say, like, it's not often that we see such consistent findings between mice and humans. However, all of the work so far in humans has been done in adolescents or adults. And if we want to use EEG as a biomarker for clinical outcomes for clinical trial, our goal is probably to do this a lot earlier than the adolescent or the adult. And so we really need to understand whether these differences are present in young children as well. And so that's what this study was aimed at. The first goal was to characterize electrophysiological similarities and differences in a group of 2 to 7-year-old boys with Fragile X compared to typically developing children. And the hypothesis there was that we would see increased gamma power in the Fragile X group compared to the typically developing group. And then second, we wanted to identify and characterize neuromarkers of verbal and nonverbal abilities. So language measures or nonverbal visual reception, those types of measures, as well as behavioral symptoms in these young boys with Fragile X. And the hypothesis there is that increased gamma power would be associated with worse symptoms. And that's largely because in Lauren Etheridge's studies, most recently their group published a rather large study with about 30 to 40 individuals with Fragile X. A lot of their findings showed that the higher the gamma power, the worse the symptoms. That being said, many of those associations were predominantly for females and not males with Fragile X. And so this is what it looks like. This is my son he's a seasoned EEG wearer in the lab. And so this is what it looks like. We measure the child's head, we place the cap on the child's head. And with the kids with Fragile X, when we do this, they're usually not sitting by themselves. They're usually in their parent's lap. We're usually using a lot of techniques, distractors, videos, sensory toys, those types of things. And we have a long discussion ahead of time with the families about what each child's unique individual needs are. To help them be distracted while they put the net on. And once they have the net on, they're allowed to watch a video. The data that I'm going to be presenting today, they watch sort of like an abstract screensaver, but a lot of times we get them first comfortable by watching their preferred video, whatever that may be. And so this is the setup for the study. Like I said, we were recruiting boys that are preschool to school aged, 32 to 84 months. And we in the end had 15 boys that were recruited. And we also had 12 age matched controls, similar aged. And then we used another set of infants that were cognitively matched, that were the EEGs were collected at the same time, but from a different study. And we collected EEG about 2 to 5 minutes, depending on the child's compliance and sort of ability to stay calm during the task. And we were able to place an EEG on the vast majority of those children. So 14 of the 15 Fragile X kids had a net placed, and we collected data. And 11 of those are shown in the, had high data quality and were used in this analysis. And then the types of assessments that we were doing, one is a nonverbal cognitive assessment called the Mullen Scales of Early Learning. Another is a language assessment called the Preschool Language Scale. We also did the Autism assessment called the ADOS, and then several parent questionnaires that were really focused on similar questionnaires that are used in clinical trials. I'm going to present, these are data that are unpublished but are recently posted on medRxiv. So if you have questions or want to look back at the data, you can search my name on medRxiv and hopefully you will find it. And so what do the participants look like? So here you can see that as expected, the Fragile X individuals had a much lower nonverbal quotient, so their nonverbal cognitive abilities, much lower preschool language scale. The Vineland communication standard score was also lower, and their irritability measure on the ABC, a commonly used parent questionnaire, was elevated. And then here you can see the group that we had that was cognitively matched. They were lower in age, but their age equivalents, so their estimated age equivalents on their visual reception and fine motor, were very well matched to our Fragile X kids. And so what did their power spectrums look like? So this is showing their power. The Fragile X kids are in blue, and then the age matched are in orange, and the green is the cognitively matched individuals. And these are just showing the power spectrum from different regions of the brain: frontal, central, temporal, and posterior. And it's really quite striking how similar it is to what we've seen in mice and in adults, that there is a clear increase in the power in the higher frequency bands, really across the beta and gamma spectrum. We did some statistical analyses using cluster permutation analyses to sort of define exactly where the statistically significant differences are, and they fall sort of in the 20 to 25 hertz all the way to 55 hertz range, and similar across all of the different regions. And so before going on, one of the things that we noticed in the power spectrum is that there's a clear increase, but there also sort of is almost like a different curvature shape. And that gets at sort of how we think about power and the different ways of measuring power. And so when we measure what I would say gamma power ranging from between the 30 and 50 hertz, what we usually do is we're taking the average of the power measured across this whole frequency window. And we do that for both groups. However, work by Brad Wojtek's group has really highlighted, and many, many others, but I think his work has really highlighted some of the problems in doing it this way. And that is that you can see differences when you average across this frequency band for many, many reasons. One could be an actual increase in the oscillations in that range. This is an example for alpha power. But it could be that there's like a change, a shift in the peak of where those oscillations are happening. And so if you're just measuring over one band region and the peak has shifted, you may not capture that. Another difference would be if the whole power spectrum has shifted up or down, or if the slope of the power spectrum has shifted up or down. Those will all give you differences that numerically may look the same, but the biological reason could be different. And so here you can see that what you can do is you can calculate the curve, we call it the aperiodic 1/f fit of the power spectrum, and you can divide up your power in a given frequency band by what we call the aperiodic component, so the part below this 1/f fit, as well as the periodic component, which is this part above it. And there's evidence that the part below the 1/f fit may represent noise in the system, or it may also represent the balance between excitation and inhibition. And the part above that 1/f aperiodic fit is more likely to be representing actual oscillations that are occurring in that frequency range. And so we can use, the Wojtek lab has developed this method called FOOF, fitting oscillations in 1/f, that estimates that 1/f curve and allows you to look at both the part of the signal below and above that 1/f. And so what does that look like for these, for our Fragile X group versus the age-matched and cognitive matched groups? So what we see is actually that there are differences still in both the aperiodic component on the left, as well as the periodic component. So this, I call it aperiodic adjusted power spectrum, is sort of subtracting out the aperiodic component from the original power spectrum and seeing what it looks like. And so what we see is that there is what seems to be a decrease in the slope of the Fragile X participants. It's not significant, but the trend is there, and you can see that that's more clearly affects the higher frequency bands. And we also see this larger peak around 30 hertz that's both shifted in frequency from the controls and higher in amplitude. And so when we recalculate what is gamma power, both the aperiodic component and the periodic component, we see that the Fragile X kids, Fragile X group has significantly increased gamma power for both of those components, both the aperiodic component and the periodic component, and significantly so. And so overall, the first goal, characterizing similarities and differences, yes, we saw an increased gamma power that has similarly been seen in the mice and adults with Fragile X, but that is seen sort of related to a reduced aperiodic slope and this increased and shifted peak at 30 hertz. And so next we asked this, how does that relate to some of the outcome measures that we're interested in? Does increased gamma power relate to worse symptoms? And here's where we were actually surprised. So gamma power, the higher the gamma power in Fragile X individuals, the better their language was for this age population. So here you can see this is the preschool language scale total standard score versus their gamma power. And this was measured not the aperiodic or the periodic. This was just the total overall gamma power. And there was a very strong positive relationship. Now it's a small sample size, but it was very clear finding. And we also saw a similar relationship with the parent report measure, which is the Vineland Communication Scale standard score. We did not see that association with the nonverbal measure on the Mullen. So it seemed like it was language specific. And so then what about the aperiodic versus the periodic components? How are those associated with language? Is one more associated than the other? And the interesting thing here was that it wasn't the periodic component that was strongly associated with language, it was the aperiodic component of gamma. So that part under the 1/F curve that was significantly associated with language ability. And we also did some linear regression analyses to account for age. And that relationship remained when we accounted for age for aperiodic gamma and language. And again, there was no association with periodic gamma and language measures, especially after accounting for age. Side note, this periodic measure of gamma was actually strongly correlated with age, so that the older you were, the lower your periodic gamma. There was no association with age and aperiodic gamma. And so just lastly, we did look at other behavioral measures. These are just showing some of the ones that I think are often used in clinical trials. So the Fragile X ABC irritability score, there was no association. The sensory profile, sort of a total raw score on that, there was no association. The repetitive behavior scale, there was no association. That being said, again, this is a small sample, 11 kids, so it may be that to see associations, we need to have a larger group. And so to summarize, yes, we did see that there was a relationship between increased gamma power and symptoms, but it was in an unexpected direction. It was that the aperiodic gamma power was related to better language. And so why might that be? Well, one interesting idea is around the EI balance and that imbalance that's seen in mouse models of Fragile X. And there's been some interesting work showing that 1/F slope is sort of representative of the EI balance of the system, and that a decrease in slope and thereby an increase in gamma represents an increased EI, so more excitation than inhibition. And then you still may say, okay, well, why would increased EI, an imbalance, an increased excitation over inhibition, why would that be good, or why would that be associated with better language? Well, there's Michelle Antoine's paper from Dan Feldman's group that was published last year, I think was really exciting for many people following this work, in that they looked at Fragile X mice along with 3 other mouse models of autism, and found that in all of these mouse models, there was an increased EI ratio. But that EI ratio being increased was related to normal spontaneous firing rates. And so the hypothesis that they've put forward is that an increased EI ratio may be representing homeostatic plasticity working and recalibrating a system so that there isn't too much firing going on. And if that's the case, then maybe our findings are in line with that. And it also may be that our findings, what we're seeing is something that's related to development. We're looking at an earlier time point, and so that also may be factoring into the differences. So overall, we found that preschool-aged to school-aged boys with Fragile X showed significantly increased power in the beta-gamma range. And that was related both to a decrease in the 1/F slope, as well as an increase in shifted beta-gamma peak. And then that aperiodic gamma was associated with better language ability. And we didn't see, at least yet, any association with other clinical measures. And so what are we going to do in the future? Well, one of the questions is still, how does this relate to task and language processing in particular? So we were showing data that's really collected without any kind of stimulus in place. The children, there's no auditory stimulus, they're watching sort of an abstract moving object. But how does gamma and aperiodic gamma measured in that instance relate to a child's response to hearing language or hearing sounds or being able to habituate to sounds? And then how does that relate to symptoms? And of course, I really think we need larger and larger studies, collaborative studies that are able to take together, larger numbers of children with Fragile X to be able to really fully understand the findings, and also compare kids with Fragile X with other similar genetic disorders like tuberous sclerosis, Down syndrome, Rett syndrome, and see what are the similarities and what are the differences in how EEG findings relate to intellectual cognitive challenges as well as language challenges. And I'd like to most importantly thank all the families that participated in this study. If you guys have questions, I'm obviously happy to answer them here, but if follow-up questions, that's my contact information as well as our study email. And I want to thank some of the RAs and students that also helped out, and the many, many different groups that helped fund this project. This was really a Fragile X community effort, I would say. And so thank you.
Thank you, Carol. That was a fantastic presentation, really interesting results, and I'm sure we're going to have some questions about that. First question from Leah Wong to everyone. How accurately can the model predict the severity of autism for 2 to 3 year olds in terms of language development, verbal or nonverbal?
Actually, I'm going to defer to Carol on that one because Carol, why don't you tackle that, because the paper you published is really centrally focused on language.
Yeah, so we, this is for the infant sibling study. So we have also looked at EEG, very similar EEG markers. So both the intercept at 6 months of age, as well as the slope over time, to instead of predicting autism and predict language outcomes at 24 months. And it actually, the measures do a very good job at predicting language ability measured on the Mullen, but a big part of that was recognizing that these EEG measures may predict language differently depending on whether you are low or high risk. And so we had to include that in the model to get the best type of prediction. For language, unlike what we found with predicting autism, we did have better predictive ability when we incorporated data all the way from 6 through 24 months, rather than just using the first year of life. And so that may give us a little indication about the differences on what things may be relevant for autism versus autism symptoms versus language specifically.
So Carol, the question asked for verbal or nonverbal, but can you divide it into receptive and expressive? Is there any difference there?
We didn't divide, we didn't do predictive modeling for receptive versus expressive. We did it just using the Mullen verbal quotient. And then we also have done it for the nonverbal quotient. We're able to predict similarly well for each, but different EEG measures are statistically sort of important in the model in different ways, depending on whether you're predicting the verbal or the nonverbal quotient. The delta, you can look at the papers in neurobiology of language. Early delta and theta intercepts were important for language, specifically in the high-risk groups. And beta ended up being, slope of beta, I believe, was important for nonverbal prediction across groups.
Do you want me to read out the question, or do you want to?
Oh, well, I can read it for you from Gordon Wong for Dr. Nelson. In your patient population, what does the EEG signal look like for low-risk children that develop autism? And I'm not sure you had a group for that.
That's correct. We have a very tiny group for that. In fact, I'd have to call in Carol and Laurel, who are on the phone right now, whether or not, you know, my memory is we're talking about 3 or 4 kids. Is that about right?
Yeah, so not really enough for us to look.
Yeah. We have another study with a very large sample on a completely different project, but over the years, probably 10 or so of those children developed autism. And we're just positioning ourselves to look at this very question. But right now, we don't have an answer to this question about what the low risk who develop autism look like.
Okay. And Xinyu Zhao asks Dr. Nelson, do you see EEG patterns that correlate with more severe forms or less severe forms of autism?
Yeah, so, you know, what we reported is in the work on feature extraction that Basel did, we could predict severity, but, you know, I qualified that by saying that the pathway to autism through this infant SIB pathway tends to be a much milder variant. So it's really, we really can't address the question of severity if you think of it as, you know, IQ of 100 versus IQ of 50 or something like that. And what has not been done, to my knowledge, is to use these same tools in a cohort of older children with autism who have a huge range in functioning to see if we can see the same associations. And that hasn't been done yet.
Okay. And Leah has a question. This is a kind of general question about Fragile X. Geneticists said that methylation has no link to prediction of the intellectual level. Is that true? I would just add that methylation does indeed correlate with degree of impairment, but they may be saying that on an individual level, it's hard to predict outcomes based on lab tests because there is a fair amount of variability. Anybody want to add anything else on that?
Yeah, I mean, I would just add that I think that with the studies that we didn't do any genetic testing as part of our study, but I think the future studies, I think not methylation status, but really sort of like protein, being able to know how much protein is being made in an individual and understanding how that relates to symptoms, I think is important. From Lauren Etheridge's recent work looking at this larger group of 40 or so individuals, they found associations really driven by females. And so part of me wonders whether that's just representing which individuals have Fragile X. And so they may find that really because the females with high gamma power were associated with worse symptoms, that may just be representing that they're the individuals that are affected by Fragile X and the others are not, which is different than saying, if you have Fragile X, how does gamma power relate to outcome measures, right? They're slightly different.
Another question for Carol. Did all the Fragile X children you tested have increased gamma power between age 2 and 7? Did you see any age-dependent changes in gamma power?
Yeah, so let me just go back to, should I go back? I can say that it was, if you look at the bar graphs, I can share it again, but the bar graphs are actually quite separated. There were some individuals with Fragile X that overlapped with the typically developing both age matched and cognitive matched group, but I would say overall from the literature, it was a pretty good separation. But there were age-dependent changes, not in aperiodic gamma, but in that periodic component, where the older the children were, the lower their gamma, their aperiodic gamma. And so for me, that I was reassured by our findings that aperiodic gamma was associated with language, especially given that there was no association with age. So it seems like that's more of a, there aren't as many confounders that are possible there.
And Laura Mamunis wanted to ask you, Carol, do the mouse models show this relationship between an aperiodic gamma shift and behavior?
I know, right? I want to know too.
We have a few people from UC Riverside on the line. I don't know if any of them want to volunteer.
I'm gonna email Khalil and be like, "Look here, she'll take a look at this and see what you guys see in the." And then we're also in collaboration with Mark Baer's lab, and so I think that's one of the next steps is looking at that specifically.
Yeah, I mean, actually, Laura, what's really interesting about that is that with this project we have with Mark, they're going to look at TS mice, Fragile X mice, another strain, I can't remember, and we can do the same thing across these strains, and then in the human, we have the same disorders. So we can do this not just for Fragile X, but we should be able to do it for a couple of other disorders as well. And that's going to be really cool, because then not only we can look at whether this is something that's common across disorder or unique to specific disorder. If we see a unique signature for specific disorders.
Daman Kumari asks, were the Fragile X children in this study under any pharmacological or behavioral therapies, and would that affect your EEG results?
Yeah, there were minimal pharmacological there were minimal kids on any kind of medications. It's listed in the paper, but no major medications, things like, constipation medications. One child was on oxybutynin, but there were also similar in the typical developing group that also had sort of like melatonin here and there. The kids with Fragile X definitely were receiving behavioral therapy. They are in I would be very frustrated if they were not getting speech therapy and had IEP services. I don't think that majorly affects the findings. I think that it is something that makes it harder to, we have a new study, we got a K award to do another study in kids with Fragile X, and that's going to be a longitudinal study so that we can not just look at a one-time measure of language, but to look at improvement in language. So are there predictors that show more progress in language versus sort of stagnant progress in language? And that would sort of get at, be able to tease apart certain things related to how much therapy they're receiving. Most of the individuals were, in the Massachusetts area, and we tend to have good services here, thankfully.
And that's another advantage, I would imagine, of getting them young, is that you have the opportunity to observe subjects that are not on any medications, whereas by age 18 in the US, just about everybody's on at least one medication, it seems. Got a message, I hope this is okay. Patty from UC Riverside says that they haven't done the aperiodic gamma shift analysis, but are considering doing that. So to answer that question. Have you generated data on specificity of predictors for autism versus other neurodevelopmental disorders?
Well, that's a fascinating question coming from someone in our lab.
It's a plant in the audience.
Exactly. No, we haven't done that yet, but we're just waiting to do that, Laurel. So for those of you who aren't aware of this, we see children with Fragile X, Rett, P10, PMS, FOXG1, CDKL5, Down syndrome. Carol, that's it, right? Is that the list? I think that's the list. And we have EEG on all of them under the same conditions. So the question is, how specific are these markers? Are they specific to disorders? Or as something we've discussed before, are we detecting early on an error in brain development? And but how specific it is, we don't know. And then even within autism, there could be subtypes. So, it's embarrassing that we actually have the data to address this question, but we haven't done that yet. Thanks, Laurel.
Good question. I also had a question, Carol, specifically about the beta peak shift that you showed in your data. I was wondering if there are any hypotheses about what the network or even cellular basis for that would be. It seems to me that if you get a pronounced peak like that doesn't really exist in the normal population, that makes me wonder if you're seeing the effect, at least on a population-wide basis, of a specific kind of mutation in a particular cell type.
Yeah. So if you look at the DUP15 literature from Shefali Jesti's work, that group of individuals has a very similar peak, but at 25, it's like 23 hertz. And very sort of strong peak. And they've done some very interesting. I'm just seeing that it's not Laurel asking that question, Dr. Chuck. But they've done some really interesting work looking at benzos and inducing that increased peak with benzodiazepines. And so their hypothesis is that the 23 hertz peak that they're seeing in that syndrome is related to issues with GABAergic receptors. And so I think that one thing that would be very interesting to look at is to try to figure out what receptors or what types of circuitry would drive a 30 hertz peak. And that's, it's interesting because 30 hertz is almost universally the divider between beta and gamma. So if you're just looking at bands and you're looking at beta or you're looking at gamma, you're dividing it right at when we're seeing the peak. And so you're probably not accurately measuring that change just by virtue of historically where we've separated these frequency bands.
Yeah, I think there's actually some benefit to potentially surveying large numbers of patients with large numbers of different pharmaceutical compounds, because it's not entirely known exactly what all of the effects of some of the commonly used medications are on EEG. Some are known and some aren't. It largely depends on what generation those drugs came out in. More recently, some of these studies are done as part of the clinical development of drug classes. Mm-hmm. But that makes me wonder if really, if what you're seeing with a benzodiazepine applied to those patients, if you're thinking about this in terms of an excitation inhibition type imbalance, that kind of model, if this shift in the beta peak, the beta gamma peak, could in fact be something like a compensatory response with an increase in inhibitory transmission.
Yeah. Carol, can I say something about that first, and then you can jump in? So I think that's spot on. The other thing is that, there's a big interest in the extent to which these GABAergic drugs shift critical period closure. And so going back to the maybe an earlier question about treatment, and then I think Katie's question about, well, maybe it was Michael, how early we can see these kids. there's an issue of critical period plasticity. And so if we see them young enough, might there be treatments that can restore EI balance and close off a critical period in a beneficial way, versus you can also think of critical period closures as premature and as preventing subsequent experience from impacting development. So the whole, I think something that Carol and Takao Hench and I and some others have talked about a lot is the extent to which in the human, these EEG biomarkers can reliably be used to infer something about EI balance. And so in the visual evoked potential, we think that the downward part of the P1 component may allow us to do that. That's where we need to ground this in the animal model, because in the animal model, you can manipulate EI balance more than you can in the human. And so if we can establish this in the animal, can we then translate it to the human? So Carol, why don't you comment on this as well?
Yeah, I mean, I think that there's a lot. I think that the work that I presented today, I think gives a little more oomph to some of the findings in the mouse models, that the EI imbalance may be representative of some compensatory mechanisms. And for me, it really pushes the need to be doing some of these mouse studies in the developing mouse. And to understand changes that occur in the developing mouse, just as we need to do it in the developing human, right? That understanding how the GABA switch happens and how that relates to differences in later measures of EI balance or spontaneous firing rates, and all of those things, I think is important.
Emily Hubbard asks, for the layperson, what are the larger long-term implications of this study in terms of treatment of Fragile X? And I would maybe even just expand that to say, what are we left with? What are the implications of this for practical utility, at least at the moment?
I think that there needs to be, that this is, I think optimistic that EEG can be a useful tool for clinical trials and as an outcome measure, but we need a lot more work in understanding what the measure represents as far as its relationship to the outcome that you're looking at, and that needs to be done at different ages. My assumption is that we want to be able to treat Fragile X as soon as possible, and we're able to. It's a genetic disorder, so you're able. If we had the effective therapy, it would be part of the newborn screen, right? And you would be able to diagnose it at birth and provide therapies at birth. And so, but that only happens if you have an effective therapy. You don't add anything to a newborn screen. You only add things that you can do something effective, have an effective therapy for. And so that's why I think we need to push a lot of our research into the younger ages, because there's a lot of utility in understanding the biology there and understanding the therapeutics there. That isn't to say we shouldn't also look at adults, because obviously we also want to be able to have therapeutics for adults with Fragile X, but they may be different. They may not be the same, and what works in an adult may not work in an infant or a toddler. And so we cannot think of them as the same thing. We really have to think about both age groups differently.
A question from Kagistia, and if it's the same one I'm thinking of in Singapore, another FRAXA fellow, my question is whether the presence of seizure in other neurodevelopmental disorders like tuberous sclerosis will affect the EEG result.
That's a great question. So when we first started work on TS, this was a big issue because, the vast majority of these children have epilepsy. So it turns out that it doesn't impact what we see in two ways. One is if there's say, well, if the child has a seizure, then that's different. We're not collecting real data, but good data. If there's subclinical activity, that can be spliced out. But as I say that, it's a little disingenuous, because just because we can sort of create a window or an envelope around the epileptiform activity doesn't mean the rest is unaffected. It just means we probably may not see it. But in the visual evoked potential, it doesn't really seem to matter. And in TS in particular, one thing that we've been puzzled by for years is not only how normal the visual evoked potential is, but that it's even a little bit more normal than normal. I know that sounds odd, but what I mean by that is it's bigger. The amplitude is larger. The latency is exactly the same. It looks, if you saw the morphology of the VEP, it would look super normal. And it doesn't, we see this in individuals independent of whether there's epileptiform activity. Now, when I say epileptiform, that's also, I'm misspeaking there, because the children are not, we know when they're having a seizure, but what we're more concerned about is that there's subclinical seizure activity going on, even if there's nothing to show this in behavior. The EEG can be impacted, but the VEP doesn't. It's as though we can average that right out. Probably that's what's going on, is we're averaging it out. I realized that was a longer-winded answer to what I thought when I read the question would be a straightforward answer. Because now I'm convincing myself we haven't looked at it as carefully as I thought we should have looked at it. This is the danger of giving talks sometimes. You realize that you're not as smart as you thought. So I will get back to you. If you follow up with an email, I'll give you a more thoughtful answer.
And I guess too, where you have epilepsy, you usually have anti-epileptic medication.
Correct. I mean, all these kids are on anti-epileptics. Now, it's less of an issue for other populations because of that list I gave you before. You know, not all those children have epilepsy or even seizures. The Rett girls will eventually do that. But, you know, the real question is when we go through an artifact score EEG, it's one thing if we're detecting movement. It's another to detect a seizure. And how legitimate is it to splice that out and then just stitch together the artifact-free epochs if we know there was background seizure activity? And that's the harder question. I don't think we've been as systematic as I'd like to think as we've been there.
I'll just add that it may not be the right thing to remove also, right? When we think about EI balance,
Yeah,
that's also related to risk of seizure. And so, and we don't do clinical EEGs on everyone that's at risk for a seizure.
right.
Probably many individuals that are never diagnosed or never have seizures, but have underlying EEG abnormalities that may be related to the different behavioral challenges and cognitive challenges, right? And so we don't necessarily want to take that out. It's interesting, I was talking to a friend of mine who's now at a biotech company and interested in kids that have seizures and have neurodevelopmental disorders, and saying, like, why don't we do genetic testing on all of those individuals? Because we may find a lot more genetic association with genes that are usually associated with refractory seizures, with really bad seizure, seizure syndromes. But we only test the kids that are doing really poorly, where it could be a much larger portion of kids that have mutations in those genes, but don't actually have epilepsy. They just have one seizure, once. And so there's just still a lot to be learned.
Well, I think that takes care of all the questions we have for now, but I'd really like to thank our speakers for really fabulous presentations today. I think this will be an all-time favorite on the FRAXA website. So it's really great to have you present this important information.
Great.
Thank you.
Thank you for the opportunity.
Yeah, same here. This was really wonderful.
Thanks very much. Well, everybody, tell your friends and neighbors and lab mates about our webinar series. There are more to come.
Good. Thank you, everybody.